Thank you. All right, we ready? Got the coffee going? All right, I'm going to try to get rolling here. So the next couple lectures we're going to do are fairly dense. So I don't have a ton of extra time here. Most of this is, I believe, is stuff you've seen before. But today we're going to talk about transcription. Once again, the objectives are there for you guys to use as you see fit. So there's our central dogma again. And yesterday we talked about the structure of DNA and RNA. We talked about DNA replication. The nice thing is, if you guys have looked at the DNA replication stuff, or you're still sort of familiar with it from my lecture yesterday, a lot of what we saw is repeated in transcription. So transcription, in many ways, looks very much like DNA replication. Certainly the synthesis of RNA is very similar to the synthesis of DNA. And then actually in the next lecture, we're going to talk about translation. So some general features of transcription. You'll notice in a few places in this particular PowerPoint, rather than using the textbook figures, you'll see these black and white figures I have. You'll actually get these figures eventually. These come straight out of the Kaplan Review Book. This is actually an older version, so the Comlex Review Book. I liked it because it was pretty simple, so that's why I picked some of these figures. But anyway, it's got the same information. In some cases, it's simplified it a little bit. Transcription is going to begin with RNA polymerase binding to a promoter region. So instead of having those origins of replication, now we have some kind of promoter region. We're going to have to kind of do a similar thing, though. RNA polymerases, and you already know this from yesterday, do not require a primer. So you've already seen primase. That was an RNA polymerase. Didn't require a primer. Unlike DNA polymerases, which do, DNA polymerases can only add on to something that's already there. So these RNA polymerases don't require a primer. So transcription doesn't require a primer to begin. The polymerases synthesize 5' to 3', just like DNA polymerases. Okay? All RNA and DNA polymerases synthesize in that direction only. The RNA polymerases use only the DNA template strand for transcription. So we're going to have, there's going to be two DNA strands. We're going to call one the coding strand and one the template strand. The coding strand is actually going to be identical to the messenger RNA that we're going to produce in terms of the sequence, except you're going to have U's instead of T's. But the template strand is actually what's copied. Okay, and as we go through this, hopefully that makes sense. So it's going to read the template from 3' to 5', so this white bar right here, and it's going to synthesize 5' to 3', just like what happened with DNA replication. And the template strand, just like with DNA, is anti-parallel and complementary to the RNA being synthesized. The coding strand, now in this figure they also use the term anti-template. I don't like that term. It's confusing and ambiguous, and I don't see it used very often. So we're just going to say coding strand. I don't think you're ever going to see this word anti-template. If you do, now you know what it means. But the coding strand, once again, it's going to be identical to the messenger RNA being produced. RNA polymerases use ribonucleotides. DNA polymerases use deoxyribonucleotides. Okay, but it's a similar process. And by convention, genes are described by giving the sequence of the coding strand. So if you go out and you read a paper, you're interested in some particular gene, or somebody did a sequencing project or something like that, and they're showing you a sequence of the gene, usually rather than show you the RNA, which is what's actually translated, they'll give you a DNA sequence. And the DNA sequence they're giving you is the coding strand. So it's the same as what's going to ultimately be the messenger RNA, except you have U's instead of T's in the RNA. But that's just by convention. So they always, well, I shouldn't say always, but most of the time when you read journal articles, they'll give you a DNA sequence. And the DNA sequence they'll give you is the actual coding strand. Okay. Okay, so we have this interesting numbering system. So the base sequences, and it's the sequence in the DNA itself is actually numbered by giving the first base that's going to be transcribed, the very 5' end of what's going to ultimately be the messenger RNA, is given a +1 designation. That's going to be the very first nucleotide that's transcribed. Okay, so if you go in the direction of transcription as the RNA polymerase is going to move along, we're going to have these plus number designations. So as we go in this direction when we're transcribing, now we're copying this template strand, we're making a identical in terms of the sequence to the coding strand, and we're going in this plus direction. Okay. Now the sequences to the left in this figure, where you can see where, for example, where this promoter region is, have these minus designations. So in this numbering system, there is no 0. You either got +1 or -1. There is no 0. Okay, and the -1 is also called upstream, and this becomes important because these are promoters, where our promoter sequences are. Okay. And so as we go through, we're going to talk about some promoter sequences, and you're going to see we're going to use minus number designations to say where they are. These sequences, anything with a minus in it, doesn't end up in the messenger RNA. Now it's important for transcription because it's in this promoter region. It kind of defines how transcription is going to proceed, but it doesn't ultimately end up in the messenger RNA itself. Only from +1 on ends up in the messenger RNA itself. Transcription ends when a termination signal is reached, and we're going to talk about that. This is just a repeat. Template strand is identical to, or RNA is copied from the template strand and is identical to the coding strand. Okay, so we're using the template strand as a template, and what we synthesize, that RNA molecule, is identical in sequence to the coding strand. And that just goes back to our base pairing rules that we learned yesterday and the anti-parallel designation. So we've already talked about that. Complement, yep. So this was all. So the only other information here is, so using the Watson-Crick base pairing, so what we talked about yesterday, A base pairs with T, G base pairs with C. With the exception, once again, when we talk about RNA, you have to replace U for T. If I give you either a coding strand or a template strand, or if I give you an mRNA sequence, you should be able to determine the other two. Okay, and you just work through the same sequence of events that we talked about yesterday when you try to determine complementary sequences. The only thing I mentioned here is pay attention to the orientation. That means designate your ends so you don't get confused where you are or whether you've written a sequence forward or backwards. Because if we do have something like this on an exam, we're going to have multiple choice answers, and the ultimate answer could be written in the forward or backward direction. You know, I'll designate the ends. If they're not designated, then you know it's written in the forward direction. Okay, so I don't want to. If you go through this, and they've actually went through the process with you here. You can start with the coding strand and make sure you can figure out how you got that template strand, how you got that mRNA. It's pretty simple stuff. If you do struggle with it because you haven't seen it before or you never really got it the first time, come see me, because I think in a couple minutes we can have it straight in my office. Okay, don't spend hours. You know, it shouldn't take you that long. Okay, if you're struggling with it, come see me. Okay, so first thing we're going to talk about is prokaryotic transcription. And in prokaryotes, you only have one RNA polymerase, so it's just RNA polymerase. In this case, it's a prokaryotic version of RNA polymerase. When we get to the eukaryotes, you're going to see, unfortunately, that's not the case. There's multiple RNA polymerases. So we have this example here of a holoenzyme, which contains both the core enzyme and the sigma factor, and we're going to define what each one of those is. Together, they make the holoenzyme. And before transcription begins, that's how it's going to exist, as this holoenzyme. We got our core enzyme, got 2 alpha subunits, a beta and a beta prime subunit. So it's a multiple subunit. So it's got quaternary structure, for example. And this core enzyme is the RNA polymerase. Okay, it's got the RNA polymerase activity, which is a 5' to 3' polymerase activity. No proofreading in this enzyme. None of the RNA polymerases have proofreading activity. So what is this sigma factor that's on here when we have the holoenzyme? That is required to recognize and bind promoter sequences. So before the RNA polymerase is bound to a promoter, when it exists in the cytoplasm of a bacterial cell, you have this holoenzyme. The sigma factor is what's going to recognize the promoter region on the chromosomal DNA and align that RNA polymerase up correctly so it begins transcription at the +1 nucleotide where it's supposed to start. That's its job. Okay, recognize, bind the promoter, and line up the RNA polymerase correctly. Therefore, you actually are going to have different sigma factors for different promoters. So certainly certain genes you want turned on at different times, right? This is one of the mechanisms by which you can control that. You can have a different sigma factor to recognize different promoters. Shortly after transcription begins, that sigma factor is actually going to fall off. Its job is actually complete at that point, and the enzymatic activity, the RNA polymerase activity, is actually contained in the core enzyme, so it no longer needs that sigma factor, it dissociates. Okay, then it can bind another core enzyme and direct it to transcribe whatever genes need to be turned on at that time. So to initiate RNA synthesis, by the way, when we're talking about the models that we're using right now, we're really talking about messenger RNA synthesis. So some of the core promoter regions and stuff we're talking about messenger RNA. You do transcribe ribosomal RNAs and transfer RNAs, and we'll talk a little bit about those, but the promoters are going to be different, and we're not going to talk about those. The promoters that are real interesting to biologists tend to be the mRNA ones, because they're the ones that encode all these different genes. Ribosomal RNAs and transfer RNAs are just turned on all the time. They're always being produced, so they don't have some fine control mechanisms. Okay, so first we're going to look at this core promoter region. So these consensus sequences, these are sequences that would be common to all promoter regions for mRNAs. So the initiation of transcription is caused by recognition of promoter sequences by the holoenzyme. So it's the sigma factor, right? It's actually going to recognize these consensus sequences. And in the promoter in prokaryotes, you have these 2 consensus sequences. When I say consensus sequences, these are present in virtually all mRNA promoters, okay, all promoter regions. And so these 2 consensus sequences are first a TATA box. Okay, this is a sequence of about 6 nucleotides. You don't have to try to memorize the sequence. There's no particular sequence I'm going to ask you to memorize, or there's no sequence, unless I specifically tell you you have to memorize, that you actually have to memorize. Now, the name kind of tells you part of what the sequence is anyway, right? So it's pretty close to being what the sequence is. And so one of the things we're going to have to do for transcription, just like when we replicated DNA, is we're going to have to open up that DNA, right, to begin transcription. So in the origin of replication sequences, if you remember, we had a lot of what base pairs in there? AT, right, with low hydrogen bonding, much easier to separate to begin. And so that serves a similar purpose in this promoter region. It's going to allow that region to be separated so the RNA polymerase can begin its work. Once it starts, just like with the DNA polymerases, it has no problem peeling a nucleotide at a time apart, or base pairs at a time apart. But to separate that initial region, it's a little harder, so that's why you have AT base pairs. Okay, it's centered somewhere around -10. So once again, you'll see in these promoter regions where we have these negative numbers. Once again, these sequences don't actually end up in the messenger RNA. They're telling the polymerase where to begin. It's going to actually begin at +1. So this is going to help open up the DNA to begin transcription and also line up the RNA polymerase. And then we have a second consensus sequence, this -35 sequence, present in virtually all genes in the bacterial chromosome. And it's centered about 35 nucleotides upstream, so about -35, that's where its name came from. So these two sequences line up that RNA polymerase so that they can begin at the correct spot. Now, these are part of what's going to be recognized by the sigma factor. Now, there's obviously different genes you want turned on at different times, so there's going to be more to the promoter than that. But this is common to all the promoters. Okay, it's the specifics of different promoters we're not going to have time to get into here. We're not going to look at very specific genes in very specific regions. Just realize those have to occur in order to have some kind of differential control. And we'll talk a little bit about that tomorrow. So elongation, so transcription begins at the +1 transcriptional start site. And as transcription begins, that sigma factor is going to fall off fairly quickly within a few nucleotides after transcription begins. And so this process of elongation, first of all, you had to open up the DNA to begin with, and then as it moves along, it creates this bubble, just like a bubble that's created during DNA replication. That's where the two DNA strands are actually separated. Now, as you can kind of tell in that picture, you don't really need, because you're not opening up a huge region, you don't really need the SSBs in this case to hold it apart. That can be held apart just by the part that needs to be held apart can be held apart by the polymerase itself. Okay. Now, just like with DNA replication, as you move along, you can create supercoils during transcription. Okay, very similar to what we did during DNA replication. It really, whether they're going to be significant or not, depends on the length of that particular messenger RNA. In eukaryotes, you can get some absolutely enormous primary transcripts. So, and I'm going to explain a little bit about transcription in eukaryotes. There's a whole bunch of stuff you got to chop out of the messenger RNAs. So at the beginning, they can be pretty huge. In those cases, you may need a topoisomerase to relieve the supercoiling. In many cases, the supercoiling doesn't get, you don't move far enough along where it gets significant enough to need them. But it can't, but you do have those enzymes available if they're needed. This should look very, very familiar because we had almost this identical slide yesterday, except we were looking at DNA replication instead of RNA transcription. But it's the same exact thing. You know, the template strand is going to define what's the next nucleotide. The next nucleotide is going to come in as a triphosphate version. We're going to cleave off that pyrophosphate, which is in blue here. And that pyrophosphate, which is not shown on this figure, is actually further cleaved into two molecules of inorganic phosphate that drives the reaction forward. Okay, and that's the polymerization step. And that's as complicated as you need to make it. Okay, so don't make it any more complicated than that. The difference here is the RNA polymerases are not going to check their work. They're not going to proofread. Okay, so if they make a mistake, and there's something wrong with that messenger RNA, they just degrade the RNA and make a new one. Okay, so they don't do any type of proofreading. Okay, so it's going to the RNA polymerase is going to move along, and then at some point it's going to need to stop. You're not going to transcribe the entire chromosome under normal circumstances, so you're going to stop at some point. And in prokaryotes, it's a little more well understood than in eukaryotes, and there's a couple different termination events or types of termination that can occur. The first is a row-dependent termination, and as its name implies, it depends on row. And row is a protein factor that's actually going to displace the RNA polymerase. Okay, so it's actually going to be on the DNA and actually knock the RNA polymerase off. Okay, I actually like to think of it as sort of the RNA polymerase is like a train moving along, and then somebody put something on the tracks and kind of knocked it off. Okay, that's how I visualize row-dependent termination. Not that I'm encouraging you to put anything on the train tracks, but then we have a different version, row-independent termination. This one's quite a bit different. Okay, what happens here is we have a GC-rich stem loop followed by a poly-U stretch. So this is in the messenger RNA itself. Okay, now obviously the GC stem loop and the poly-U stretch in this case, is actually part of the gene. But when it gets transcribed into a messenger RNA, it will actually make that stem loop. Okay, so the stem loop we're talking about is going to end up in the messenger RNA. And when that stem loop gets formed and there's a poly-U stretch after it, that will cause the RNA polymerase to fall off. What I like to think of it, once again, trying to simplify things and visualize what's happening to make it kind of easier to remember, I kind of like to think of it as like sort of that belt loop on the back of your pants. That's what that stem loop is, and you got, you know, something on the RNA polymerase that catches that and kind of pulls it off. Okay, that's not exactly how it happens, but that kind of helps you visualize what's going on. And so here's a nice sort of summary figure that, once again, this is from the Kaplan book, and you can see here's our -35, our Tata box, or Pribnow box, centered at about -10. So there's our POTA regions, +1 transcriptional start site. That's where everything begins. We're going to make a messenger RNA. When we transcribe it, in this case, we're going to have one version of termination, which is the row-independent version, where we have that GC-rich stem loop. So you can actually see what I'm talking about there. You get some GC-rich sequence that base pairs back on itself, okay, and a poly-U stretch, and that's going to cause termination. Now, ultimately, that can be translated. And here's our first look at kind of what a mRNA is going to look like. We're going to have a coding region, a 5' and 3' untranslated region. We're going to really expand on that as we go through. So prokaryotic messenger RNAs can actually be polycistronic. You don't see that in eukaryotic messenger RNAs. So what do I mean by polycistronic? It means you can have a single messenger RNA that encodes several different proteins. Okay? So, and that's what's loosely shown in this figure here. We got 3 different genes. Usually in prokaryotes, what it is you have multiple genes from the same biosynthetic pathway, and it kind of simplifies the way they regulate their genes. Okay, if you need this biosynthetic pathway turned on, instead of turning a whole bunch of individual genes on that are all needed for that pathway, it makes a polycistronic messenger RNA, which encodes multiple genes in that pathway. So we'll see an example of that tomorrow. Okay, but that happens frequently in prokaryotes. In eukaryotes, that doesn't happen. It's always a single gene in a single messenger RNA. Why is that? Well, when we look at how these messenger RNAs are translated, you will see why you can't have a polycistronic messenger RNA. You can certainly make one in the lab. We can easily make one and stick it in a eukaryotic cell, but what will end up happening is we'll only translate one protein from it. And I'll show you why that is later. Okay, here's our second example of a particular antibiotic at least that I've talked about. Get this through my microphone, sorry. So this is rifampin or rifamycin. And yesterday we talked about the quinolone antibiotics, which were those floxacins, like ciprofloxacin. So this is our second one, rifampin. So this is actually a prokaryotic transcriptional inhibitor. Okay? The one we looked at yesterday was a replication inhibitor that affected DNA gyrase, right? This actually affects the prokaryotic RNA polymerase. You can see in this picture it actually makes the RNA polymerase distorted so that it cannot transcribe the genes. Where you see this antibiotic used frequently is to treat tuberculosis. So I think even beginning this block, you're going to learn quite a bit more about antibiotics, and certainly by the end of med school, you're going to know more about antibiotics than you ever wanted to know. So I'm giving you some of the major classes of antibiotics to kind of give you a foundation. We're going to see a couple more today as well. And it covers quite a lot of the groups, and then you're going to get in more detail later on. So for right now, that's the level of detail I want you to know, and the story will be expanded upon later. Okay, so now what we were talking about was the transcription of messenger RNA molecules. Now the synthesis of tRNA and ribosomal RNA in prokaryotes is going to be very similar to what happens in eukaryotes, and we'll look at what happens in eukaryotes real briefly in a few slides ahead. Okay, so the prokaryotes basically just do the same exact thing, so we don't need to have two different models. The only difference is that in prokaryotes you have this one RNA polymerase that does everything. In eukaryotes, unfortunately, for you guys, that's not the case. That means we have a couple more enzymes to learn. So now we're going to look at eukaryotic RNA synthesis and kind of compare and contrast with prokaryotic version. So first we're going to look at some DNA structures. Remember that eukaryotic DNA is highly condensed, or can be highly condensed at least. And in order to allow genes to be transcribed, some of these must be exposed. So we have to, that structure we talked about a few lectures ago, you have to have it in an exposed state, which for eukaryotes happens to be that beads on a string model. You don't have to make it completely naked. Once you get down to the beads on a string model where you just have the nucleosomes, that DNA in that form is able to be transcribed. But what it means is that when we look under the electron microscope, that's what that's supposed to be right there. You can't see this with a light microscope, but under the electron microscope, you can see in the nucleus some different regions. You can have euchromatin, which is what's shown kind of in here, where it's more white. This is the less dense, and this is less compact DNA. It's able to be transcribed. How does that happen? Well, one of the things that can happen, we didn't look at real high order structures, but we certainly talked about nucleosomes, and we talked about adding or removing histone H1. When you add it, it makes the nucleofilament, right? And when you remove it, it goes back to that beads on a string, those free nucleosome model. So if you acetylate it, you can actually revert to nucleosomes. So what the acetylation does, as you might imagine, we talked about histone proteins. What kind of charge do we have on histone proteins? Positive charge, right, to bind negatively charged DNA. Well, what acetylation will do is neutralize some of those positive charges, so therefore it doesn't bind DNA as tightly. Okay, and conversely, you can actually remove or deacetylate that H1 if you want it to be bound again. Okay, so that's how we can bind and remove H1. Whereas heterochromatin, which is these darker regions, the nucleolus right here is the real dark region. Those are our little ribosome factories. So I'm not talking about that. We're talking about these dark regions right here. That's heterochromatin, and that's going to be DNA, it's highly methylated DNA that's not actively being transcribed. Okay, it's in a very condensed structure. It's not available for those RNA polymerases. So there are 3 eukaryotic RNA polymerases, and unfortunately, yes, you do need to know these 3. One warning I'll give you is, right now we're talking about RNA polymerases, and we're talking about eukaryotic RNA polymerases. Unfortunately, the names sound very similar to the prokaryotic DNA polymerases, but they have nothing to do with each other. Okay, so because we had our prokaryotic DNA polymerase 1 and 3, right? And so we've got a 1 and a 3 for the eukaryotic RNA polymerases. Unfortunately, they don't have distinctive names. So that's my warning, is just don't get those confused. That's one of the things people will typically get confused on an exam. So the eukaryotic RNA polymerases, there's 3 different versions of them, RNA polymerase 1, 2, and 3. Okay, so polymerase 1 is transcribing the ribosomal RNA in the nucleolus, which we just saw the nucleolus on the last slide. RNA polymerase 2, that's the one we're going to talk about the most today. That's the one that makes messenger RNAs. That's the most exciting one because that's the one that's differentially regulated. So Pol 1 and Pol 3 are actually going to be going all the time, basically. And polymerase 3 transcribes transfer RNA. Now you'll notice I've given you a little bit more detail just to be complete. RNA polymerase 2 actually does transcribe some snRNAs, and I'll explain what those are a little bit later. And RNA polymerase 3 also does transcribe one of the ribosomal RNAs and some snRNAs, but that's just an aside to be complete, so that way if you do some additional reading, you don't come back to me and say, hey, Dr. Lucia, I found this information that says this. That's true. But what you guys need to know is the part that's not in parentheses here. So RNA polymerase 1, ribosomal RNA, RNA polymerase 2, messenger RNA, RNA polymerase 3, transfer RNA. Okay, that's what you would be expected to know on a board exam or for my exam. Okay, once again, so we're still talking about eukaryotic RNA synthesis. You'll notice this is going to look pretty similar to what was going on with prokaryotes. We also have consensus sequences. So RNA polymerase 2 doesn't have that sigma factor that you saw in prokaryotic transcription. It is going to have a whole bunch of transcription factors, and some of those we're going to talk about tomorrow, I believe. So it's not completely by itself. There are other things going to be involved, but it doesn't have the sigma factor. Okay. So the RNA polymerase 2, which transcribes messenger RNAs, along with some of the transcription factors we'll talk about tomorrow, are going to recognize consensus sequences in the promoter. So, and these 2 consensus sequences in eukaryotes form a similar function as the -35 in the Tata box we saw. So we have a Tata box in eukaryotes, also called the Hogness box. Similar idea. It's actually located a little bit differently. It's centered right around -25. So it's the eukaryotic RNA polymerase is actually a little bit bigger. But once again, it's rich in AT base pairs, so it's going to be able to be separated so we can open up that transcription bubble. Okay? And you also have the CAT box. Once again, these 2 are also going to serve to line up the RNA polymerase in the correct spot to begin transcription at the +1 nucleotide. So the elongation process is going to be just like what happened with in the prokaryotic model. You know, we just add 1 nucleotide at a time, same exact thing. It's a different polymerase, but it's the same exact enzymatic process. Now, in prokaryotes, we had that row-dependent and row-independent termination. In eukaryotes, termination is not well understood. Okay, and that's where we're going to leave it. That makes it easy, right? Well, I guess we don't have to learn that. It's not well understood. That's as deep as we're going to go. So there's a couple little differences there with the eukaryotes. You know, the promoters are a little bit different, but it's very similar overall. So eukaryotic synthesis of ribosomal RNA. So, and once again, with the ribosomal RNA and the transfer RNA, basically the models are going to be the same between the 2, with between prokaryotes and eukaryotes. The difference being prokaryotes use the same polymerase for all of them. In eukaryotes, you use RNA polymerase 1 for the ribosomal RNA. And 3 of the ribosomal RNAs are actually transcribed as a single unit. Okay? And we have to make these individual, so what you actually do is you're going to have some cleavage events where you're actually going to transcribe these 3 ribosomal RNAs as one single unit, And then post-transcriptionally, you're actually going to modify it by cleaving those 3 fragments out to make the mature ribosomal RNAs. There is a 4th ribosomal RNA. You guys already knew that there's 4 ribosomal RNAs in eukaryotes. And that 4th one happens to be transcribed by RNA polymerase III. Okay, next we're looking at transfer RNA synthesis. So RNA polymerase III in eukaryotes transcribes transfer RNA. In prokaryotes, it's just the RNA polymerase. So you might remember the transfer RNA is very small. Even in its initial state, it's very small. One of the things about transfer RNAs is we have a whole bunch of processing events that go on, both in prokaryotes and in eukaryotes. So some of these processing events, we have to remove some intron loops. Okay, so what's shown here in purple is an example of an intron loop. In the mature transfer RNA, that's not going to be there. So we actually have to cleave that out and join those 2 ends. Okay, we're going to talk a little bit more about splicing shortly. We have to actually, there's extra bases on both the 5' and 3' end of transfer RNAs initially that have to be cut off. We also have a whole bunch of modified bases. If you look at the sequence in here, you'll see some things you don't recognize, like all these D. Okay, what the heck is that? Well, those are some kind of modified base. One of the events that happens is a methylation. You methylate different bases. If you methylate uracil, you actually end up with thymine. So remember, transfer RNAs actually had thymine in there, but there's all kinds of weird bases in there. Why are they there? I'm not exactly sure what the reason is. Could certainly impart some stability because they're not conventional. They would be a little less likely to be cleaved, but some of that puzzle remains to be solved. And then you have the addition of the CCA sequence. So you already knew that at the end of all transfer RNAs, there is a CCA sequence, right? That CCA sequence, however, is not in the original gene when it's transcribed. It's added post-transcriptionally. So you have an enzyme that's going to come along and add the CCA to the end of all of your transfer RNAs. So there's all these multiple processing events that go on. Now, eukaryotic messenger RNA synthesis is actually separated both spatially and temporally from translation. That means transcription occurs in the nucleus in eukaryotes, and translation occurs in the cytoplasm. So spatially, they're actually separated. And transcription occurs before translation. So all the events in transcription, all the processing has to happen first. Then later, it goes to the cytoplasm to be translated. Now that, in prokaryotes, we actually can begin translation before transcription finishes. So there's no nucleus there. Once enough of that new messenger RNA is hanging off the RNA polymerase, ribosomes can come in and start translating. Okay? So eukaryotic messenger RNAs actually undergo some pretty extensive processing events themselves, somewhat similar to transfer RNA processing. So here is a comparison. This is kind of for you guys to kind of visualize what's going on. We've already looked over here, and you have promoter regions, 5' and 3' untranslated regions. I'll explain what those do later. Coding regions, those are common to both. What you'll notice in eukaryotes, you have this poly-A addition site. You also have introns and exons in there, so we're going to have to do some splicing, as I'm going to explain. You have some unique 3' and 5' termini on the transcript. The 5' cap's not shown on here. We're going to see that in 1 second. We have a poly-A addition. So for eukaryotes, we have these multiple processing events. First one we're going to look at is the 5' capping. We looked at the structure yesterday, so you already knew that there was a 5' cap on eukaryotic messenger RNAs. This is a post-transcriptional event. Okay, so, and actually if you look at it up close, I'm not going to ever ask you to draw this out, but it's actually a very weird linkage. So it's actually linked backwards. So if you were going to continue on and the appropriate things were available, you would actually transcribe in this direction. So you can almost, so that 5' cap is actually attached on there backwards. And you know, that kind of makes sense with the way those enzymes have to work. But what I really want you to know, first of all, I want you to know that that the 5' capping only occurs in eukaryotic messenger RNAs. I want you to know what that cap does as well. Why is it even there? So it's got a couple things. First of all, it's a ribosome recognition signal. I'm going to show you in the next lecture how eukaryotic translation begins. But one of the things that's going to happen is the small ribosomal subunit is going to recognize that 5' cap to begin the scanning process, which I'll explain. The other thing is it actually stabilizes the messenger RNA a lot. I told you that RNAs happen to be very labile, right? They get broken down very easily. Even your best RNAs usually have a lifespan on the order of hours. And most of the enzymes that break those RNAs down are exonucleases, so they act from the end. And so this 5' cap actually protects the end, at least for a period of time. You also have the poly A tail. One thing about the poly A tail to recognize is this is not encoded in the gene. Okay, this is something that's added post-transcriptionally to messenger RNAs. So you come in and there's a poly A polymerase that actually adds the A's at the end, and it can add a variable amount of those. It can make it longer or shorter, which can be important. So what's the function of this poly A tail? Well, the poly A tail actually, it does have a function in translation. Okay, so it enhances translation, but it also serves as a signal for transport out of the nucleus. This means when that poly A tail is there, that means, okay, now the processing events are completed. You can transport that messenger RNA out so it can begin translation. Also stabilizes the end of the messenger RNA. Once again, the nucleases, many of the nucleases that act on the RNA molecules are exo, and they act from the end. So you have that poly A tail there, it can start chewing away and still not completely render the mRNA useless. So this is a way in which you can actually vary the stability of an RNA. If you want to make it more stable, you can add a longer poly A tail. If you want to make it less stable or last less long, you can make a shorter poly A tail. So the length of the poly A tail imparts a relative stability or instability to the molecule. Okay, so we have to have a number of processing events beyond the 5' and 3' ends. So I'm going through this. All of these things are basically taking place at the same time. I'm kind of walking you through a sequence. So I don't mean to imply that the removal of the introns is the last thing that happens. Really, the last thing that should happen is the addition of the poly A tail, because that's going to signal it's time to send it out of the nucleus. But all these things are kind of going on at the same time. So earlier I said there was introns in eukaryotic messenger RNAs. These are sequences that are not expressed. They're intervening sequences that need to be removed. Why do eukaryotic messenger RNAs have these? The short answer is I don't know. Now there are hypotheses out there. I don't know that anything's been proven conclusively, but we do know that some messenger RNAs are the intron regions are way, 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 way bigger than the actual expressed sequences. So the introns that get cleaved out are enormous, which is very odd. Okay, it seems very odd if you think of biology the way that I do sometimes in terms of efficiency. Why would you do that? Why would you have all this extra DNA and all these, you're going to waste all this RNA? But it does. And you have these introns that need to be cleaved out, and you have to join the exons, the expressed sequences, together. Ultimately, a mature messenger RNA is only exons. Now, one of the things you're going to see is having introns allows us to splice exons together differently, so we can actually get multiple genes from one transcript. So that's certainly a benefit of having introns. And there are some others we may have a chance to talk about. So there are some good ideas, but why they're so enormous, I'm not sure. So we're actually going to have to splice those out, and that's done by SNRPs, SNRMPs. So this is composed both of proteins and these snRNAs. I told you, we mentioned that earlier. This is another class of RNA. We didn't group it in one of the three major classes of RNA, so there are a few other types of RNAs. These RNAs are grouped with some proteins to form the SNRPs, which are basically going to be our enzymatic cleavage scissors, these molecular scissors, which are going to combine with the primary transcript. We're actually going to cleave out the intron and join the exon together. So we actually have to have two cleavage events and then a ligation event. And the SNRPs have the enzymatic activities to do that. When they're all joined together, they'll be called the spliceosome. This is when you have the SNRPs combined with the primary transcript, that's the spliceosome. We're just trying to get you familiar with some of the terminology you might see. The laureate is actually this intron that gets cleaved out, which looks sort of like a rope, so that's why they call it the laureate. Now, these splice sites have very specific, we're not going to look at the specific sequences, but they are very specific. They're specific sequences that say this is a splice site. What that means is if you have a mutation in one of those, you can get incorrect splicing. So there's another type of mutation that we'll eventually see some diseases that have that. Then the exons can be joined together. You can have anywhere between 0 and 50 introns. Most genes do have introns, and one of the things we do know now is that most genes also undergo alternative splicing. Okay, so there are, and what I mean by alternative splicing is we can actually join exons in different ways. You could have a messenger RNA here where exon 1, 2, and 3 is there. In this case, we can have an alternative one where we actually cleaved out exon 2 as well, or any number of different events. And the latest that I saw was they think about 95% of eukaryotic genes actually undergo some kind of alternative splicing. So that helps explain some of the prevalence of introns. So you'll end up with proteins with similar domains, or they'll have common domains, but they have different functions. They function differently in different cells. They may do things a little different. So you can sort of join different exons together, and it'll actually end up having, and that actually allows us to get multiple different genes from a single DNA sequence, get multiple different protein products from a single DNA sequence. But surprisingly, humans had much fewer genes than we originally thought, and this kind of explains part of the reason. Okay, I have a couple clinical correlations for you. The first is lupus. This is an autoimmune disease that you guys will expand on a little bit later, but how it ties in to what we talked about today. Some people with lupus will actually produce antibodies that recognize nucleic acids, including double-stranded DNA. Very weird. To me, it's weird to have an antibody that recognizes something like double-stranded DNA. I always think of antibodies as recognizing proteins, for example. But you can have antibodies that recognize double-stranded DNA. That happens in lupus. You can also have antibodies that recognize host proteins. So this is an autoimmune disease. You're producing antibodies against yourself. Some of the antibodies produced against yourself could be against SNRPs. Okay, so the splicing machinery. And this is just showing you the characteristic. This is very characteristic of somebody with lupus. Lupus also typically strikes relatively young adult women, which you can also see in that picture. So you'll expand upon the story of lupus. Another thing I want to introduce is the death cap mushroom, Amanita phalloides. Okay, it produces this toxin, alpha-amanitin. Now I know Dr. Danilishan, I don't know if he's talked to you yet about it, but there's another toxin you'll see in there, phalloidin, that binds to actin. But the real danger in these Amanita mushrooms, they produce a whole bunch of toxins, but the one that's going to kill you is this alpha-amanitin. And so they say as little as half of one of these mushrooms will actually kill a person. And what it does is that toxin actually specifically binds to RNA polymerase II, which makes what? Messenger RNAs, right? And the way your body works, so if you eat this, your liver pretty much gets first crack at everything. And so it's going to get the big dose of that alpha-amanitin. It's going to inhibit messenger RNA production. And basically, you're going to completely shut down your liver and destroy it. So in the case of Amanita poisoning, really the only cure for this is to have a liver transplant. Okay? So when you're studying this, I want you to know alpha-amanitin, and I want you to realize the specific RNA polymerase it inhibits, and you should also know that that RNA polymerase II makes messenger RNAs. Okay? All right. So I'll see you guys in a couple of minutes. I think that's the last slide. This next slide was just, that's an FYI slide for you guys to study from.
I have one question. So for the tRNA should have a T base pair, right?